Dr. Alzheimer's microscope slides of the plaques and tangles in autopsied brain samples confirmed his clinical observations of patients losing their short-term memory as the plaques polluted the brain's memory-forming process. Researchers tried for 80 years to analyze and understand how the plaque substance functioned. And in 1987, a team of research scientists broke the chemical code for the substance and identified it as a protein-based beta amyloid.
And beta amyloid is the building block of those plaque lesions. And so that's where we were in '87. We knew that it was the building block, but we didn't know where it came from, how it got there, or why it was produced, or why some individuals had a lot of it and others had almost none. And so that's where we started. And over the years, over the past 15 years, this beta amyloid material has become the villain, if you will, of the disease that we've been trying to conquer and trying to get rid of. It's very akin to, say, cholesterol in heart disease. The feeling has been, and the belief has been, and the hypothesis has been, if we get rid of that, and we get rid of the plaques, that these patients will be benefited from it. And that perhaps, hope beyond all hope, they'll actually be able to think better again and be able to do normal functions again if we can get rid of this material. So that's where we started. And over a series of years, we made steps and milestones along the way. First, we understood how the peptide is made and biochemically, and then later we actually were fortunate enough to develop a mouse model of the disease process. And what that is specifically is there's a mouse, and we call it the PDAPP mouse, but there's a mouse that has a defective human gene for Alzheimer's disease. And as a result of this extra gene that's been put into it, as these mice age, they get the very same plaque lesions that are seen in the brain tissue of Alzheimer's patients. So that was in the mid-90s that we achieved that goal. And it was a key goal because once we had that mouse, we could then ask, gee, if we have an idea about a given treatment, maybe we could give something as simple as aspirin or ibuprofen or whatever it is we thought we should give, we could give to these mice. And at a later time, after we had treated them for a while, see if we had done any good, see if we had reduced or removed the pathology, these plaque lesions. And so it was in these mid-90s that we were brainstorming about various things we might want to try. And one idea that I had during that time was actually extremely simple, and that was to vaccinate or immunize these mice with the very problem, beta amyloid peptide itself. And the idea was that if these mice mounted an immune response against beta amyloid peptide, it actually might help them out. It might reduce the plaque lesions and the plaque count and the numbers and that sort of thing. Now you might say, well, that is a simple idea, so did you jump on it right away? Well, no. And the reason is that curiously enough, as neuroscientists, we're taught that the immune system can't get into the brain, that the brain is protected from the immune system and from antibodies in particular. And so when I suggested the vaccination approach for these mice, my colleagues pointed that out to me. They said, well, interesting idea, but it simply won't work because the immune system won't be able, or the antibodies won't be able to get into the brain to do what you want them to do. It turns out, as a result of our previous experience working on brain and working on cerebral spinal fluid and what have you, we actually had evidence, very specific evidence, that minute, tiny quantities of antibodies do get into the brain. Very, very small. But I actually calculated that that small amount should have an effect. And the end result is, after a lot of convincing, I convinced my colleagues to try a few animals. It wasn't a big study because still no one believed it should work. To just try a few extra animals, these special genetically altered mice, tried vaccinating them and see what would happen. And that's how we finally did the first experiment. And we vaccinated them for an entire year. In other words, we wanted to ask if we vaccinated them at a young age where they had no plaques. Eventually, when we looked at their brain tissue, when they should have had many, many, many plaques, what happened? And the long and the short of that analysis is a year later when we looked, they had almost no plaque lesions at all. Almost none. In fact, so few that we assumed something must have gone wrong and we had confused the cages and we had the wrong mice. We had normal mice. And of course, it turned out in the end that, in fact, the plaques weren't there, and they weren't there by any criteria that we looked at. And over time, over the past couple of years since that discovery, many laboratories, I think 7 or 8 laboratories around the world, leading laboratories, have reproduced the result and now very much believe it. And as a result of that, it's opened up new areas of research and science. The brain is no longer considered totally immune privileged. As a result of our animal findings, we were encouraged to move on to patients, and we began that effort in the late 90s, and that continues today. What the vaccination or the immunization approach does is when an individual is immunized with A beta peptide, they generate antibodies to it. A few of these antibodies get into the brain and they act as sentinels. They actually physically bind to the plaque lesions, and the scavenger cells in the brain come along and engulf and destroy the plaque lesions. This is what we see. We have very detailed information of this occurring in our mouse animal model, and there is now some evidence of this occurring at least in one of the autopsy cases from one of the individuals that was treated with AN-1792. And so we believe that plaques are literally physically removed from the brain. It is the hope then that once these are removed, that the remaining neurons that are either healthy or mildly altered will improve their connections, their functionality, and their health, and that will allow the patient to think better. So we're just now finishing the phase 2 study, and unfortunately, when we initiated that study, we were 2 doses into that study, and this was throughout the US and Europe at something like 28 sites, 375 patients. We ran into an unfortunate side effect, and that was brain inflammation. The term for it is meningoencephalitis, and we ran into that in about 5% of the patients that received AN-1792. As soon as we saw the first signs of that, we halted all dosing. We didn't halt the trial. In other words, we kept the study fully blinded to the investigator and the patient for a full year to allow us to ask later on if indeed there might have been efficacy or whether or not the AN-1792 actually worked. But we did halt the study for safety concerns. We are now at the point of having a wealth of information coming forth with regard to A beta immunotherapy in general. And a good example of that is the autopsy case that was just reported in Nature Medicine. And in that report, an individual that had been in the AN-1792 trial for over a year eventually died to a pulmonary embolism. An autopsy was done, and to everyone's surprise, she had almost no plaques in her brain tissue in most areas. Not all areas, but most areas. The neuropathologist was extremely surprised at that result and had never seen such a thing. And the importance of that is it suggests that the mechanism that we hope is working in these patients is indeed working. If we think about the future for a moment and the possible role of A beta immunotherapy in that future, it could be extremely important. I think therapy for Alzheimer's disease is entering a new era. At the present, what we try and do for Alzheimer's victims is to bump up their cognition as much as possible, deal with their behavioral problems as much as possible. But we have no real tool to alter the deteriorating ongoing pathology in their brain tissue. And my hope, naturally, and of course, is that A beta immunotherapy will be the first of several approaches that can actually go after the underlying cause of this disease, to get rid of the plaques or reduce them. I hope that other approaches will be available as well, perhaps to go after tangles, for example, or to bolster the neuronal structure itself. What this will mean is that eventually we will be at a point where people will no longer die of Alzheimer's disease. Rather, it will be a disease that has to be dealt with, that has to be controlled, that can be treated with lifestyle changes, diet changes, and of course, therapy and medication like we're talking about right now.
The autopsy of the AN 1792 vaccine clinical trial participant is the first documentation of the plaque process being cleared from the human brain. All of humanity hopes that this is a milestone, marking the beginning of the end of Alzheimer's disease.